This is a production of Cornell University.
Okay, thank you so much, Hannah. Today, I'll be talking about genome-wide association studies for cassava brown streak disease. And for those who do not know cassava, it is a bushy shrub that is grown for its tubers around the world. It feeds about more than 800 million people, most of whom are in sub-Saharan Africa. So as you can see here, cassava is grown in different parts of the world. Don't mind the shade of the green, but I would like to bring your attention to the places in the dark green. That's East and Central Africa, where cassava brown streak disease is causing enormous yield losses that are threatening food and economic security. It also worries breeders that the virus would move to other cassava producing regions around the world, which would further increase food and economic security for people. So cassava brown streak disease is a positive sense single-stranded RNA virus. It's in the genus Epomovirus and family Portviridae. We have two species of this virus, the cassava brown streak virus and the Uganda cassava brown streak virus. Both cause similar symptoms that lead to annual losses of 750 million in Uganda, Kenya, Tanzania, and Malawi. So to evaluate cassava brown streak disease in the field, we use the 1 to 5 scoring method for assessing this disease. So the score applies to both folia and root symptoms. So CBSD folia symptoms are characterized by chlorosis along the secondary vein margins, while in the root it shows up as dark brown patches as necrotic lesions. So in the past, data from this 1 to 5 scoring method has been used in trying to identify genomic regions that are associated with CBSD phenotypes. So basically what happens is breeders score it when a score of 1 indicates there are no observable symptoms, while a score of 5 is the highest severity possible. So an example of such a study where the 1 to 5 scoring method has been used to try and identify genomic regions associated with cassava brown streak disease is a study from Zuki et al. In 2017, where they did QTL mapping for cassava pests and disease. So they evaluated cassava brown streak disease, which I'm studying, cassava mosaic disease, which is another virus disease that affects cassava across Africa and Asia. And then the pest that they studied was the cassava green mite. So they generated a biparental population that was made up by crossing a variety called Kiroba, which is a landrace in Tanzania that traces its origins back to the 1930s when breeders started breeding for disease resistance in cassava across East Africa, and AR37, which is a breeding line. So they ended up with 106 F1 progeny that were tested in 2 years across 2 locations. So they also developed a linkage map that had more than 1,500 SNPs, and I used it to identify QTLs that were associated with the 2 diseases and the pest. So I will concentrate on cassava brown streak disease. So for root necrosis, they identified 2 QTLs that were on chromosome 5 and chromosome 12. And these QTLs explain 10% of the phenotypic variance that was observed. While for the folia symptoms scored at 3 and 6 months after planting, they observed 7 QTLs that were on chromosome 4, 6, 12, and 13, and these explained 8% of the phenotypic variance. So this group went ahead to combine the root necrosis scores and the folia scores, and they identified 2 QTLs on chromosome 11 and 15. So they later concluded that there was consistency in some SNPs that had load scores greater than 2.5, but they also observed that there was inconsistencies in SNPs across locations. So that means that a group of SNPs would show up in one location, but would then show up in another location. And they also concluded that segments on chromosome 1, 12 and 13 were from the integrations that were done in the 1930s, and these came from Kiroba, which is a clone that has some level of tolerance to cassava brown streak disease. So another study where the 1 to 5 scoring method has been used to try and identify genomic regions associated with cassava brown streak disease is a study that was done by Kayondo et al. In 2018, where they did genome-wide association mapping on 2 breeding panels and tested these in 5 locations in Uganda. So they evaluated cassava brown streak disease at 3, 6, and 12 months after planting, that's foliar and root symptoms, and identified genomic regions that were associated with these phenotypes. One of these regions was on chromosome 11 for CBSD at 3 months after planting and CBSD at 6 months after planting. So SNPs on this segment on chromosome 11 explain 6% of the phenotypic variance that was observed. So another region that was identified in the study was on chromosome 4, and this was later associated to the introgressions that were done in the 1930s. These authors also reported inconsistencies in SNPs across the 5 environments. So it's important to know that the genomic regions that have been identified in both studies have never been validated. So this brings us to my research, where we're asking 2 questions. The first one is, we want to know which genomic regions are associated with cassava brown streak disease phenotypes in the cycle 2 population of genomic selection. And if there are regions that we can identify, are there common SNPs associated with these phenotypes across the seedling and the clonal evaluation trials. So to give you an insight into the cycle 2 population of genomic selection, these populations are developed based on breeding values. So we use breeding values to estimate the performance of parents that we use to make crosses. So this started with 52 clones that were selected from Tanzania, the Tanzanian breeding program, that were from the CIAT program and the International Institute of Tropical Agriculture, Nigeria. So these 52 clones were hybridized to create the C0 population. And from the C0, a subset of 100 clones were taken based on their breeding values to create the cycle 1 population. And from the cycle 1, we also got 95 clones that were hybridized to create the cycle 2, which I am working with. Now, in the cycle 2 population, we ended up with 6,700 seedlings that were planted in 1 location. And from these seedlings, we did subset 303 clones that were advanced to the clonal evaluation trials, where we planted them using augmented designs with 5 checks. We evaluated these 303 clones in 2 locations for 2 years. Now, because we're talking about genome-wide association studies, our marker data, we worked with marker data that had 41,000 SNPs that were from that sick genotyping. We did the 1-step GWAS and then we used the FDR correction threshold. So if you look at the seedling trial, which had the 6,000 clones when we did GWAS, you can see that at 3, 6, and 12 months after planting, we see the same trend, that we see numerous hits across, different chromosomes for each trait. So it's important to note that for CBSD at 3 months after planting, the SNPs that were identified explained 11% of the phenotypic variance in the seedling trial. And then at 6 months after planting, the SNPs explain 5% of the phenotypic variance, and at 12 months after planting for root necrosis, they explain 7% of the phenotypic variance that we observed. But we had asked another question, that are there any common genomic regions across these 3 phenotypes? And the answer was yes. We could see this on chromosome 1 and on chromosome 14. So we did the same thing for the clonal evaluation trial, and the same trend happens, that we see that we get numerous hits on different chromosomes for each phenotype. So you could see that for CBSD at 3 months after planting, the SNPs that we identified explained 16% of the phenotypic variance that we observed, which was much higher than that of the seedling trial. And the same thing happens for 6 months after planting, where we see 12% of the phenotypic variance being explained by these SNPs. And at 12 months after planting, it was 18% of the phenotypic variance. So the same question was asked if there were genomic regions that were consistent across the 3 time points. And here we only see 1 on chromosome 12. So we went ahead to ask yes, we've identified these genomic regions, but that there are SNPs, in the seedling trio and the clonal evaluation trio that are common across these three phenotypes. So we went ahead to check for the seedling trio. All the SNPs that we got for the 3 time points, we did not have any common SNPs across these phenotypes. And for the clonal evaluation trial, you could see that for foliar symptoms, those that are evaluated at 3 and 12 months after planting, we only have 4 common SNPs but we don't have any SNPs between foliar and the root symptoms. So we did also ask another question, is if there were common SNPs between the seedling and the clonal evaluation trial, and the answer is no, we did not have any common SNPs between the seedling and the clonal evaluation trials. So now that we don't have any common SNPs but have common genomic regions, we changed the question and asked, are there common genomic regions in our population with those that have been published? That's Nzuki and Kayondo. And here you can see that for when we compare our results to those of Nzuki in 2018, you can see that for CBSD 3 months after planting in the seedling trial, we do, and 12 months after planting, we do have common genomic regions on chromosome 4, 6, 12, 13, and 5. And then when we go to the clonal evaluation trial, for all the 3 phenotypes that we evaluated, we have common genomic regions with their population. When we do look at Kayondo's paper, you see that we only have common genomic regions for only CBSD at 6 months after planting, and that's on chromosome 11 in the seedling trial and chromosome 11 and 12 in the clonal evaluation trial. So having done this, the take-home message, you could see that chromosome 4 and chromosome 12 appear more frequently between both studies, between the 2 studies and our research. So the take-home message I would like you to take about cassava brown streak disease is, 1, we've identified SNPs, but you can clearly see that these SNPs vary across the 3, 6, and 12 months time points of evaluation. We've also seen that these SNPs vary between the seedling and the clonal evaluation trial. Even if I didn't show the data, they do vary across years. So if you test the SNPs that show up in 2019 and 2020 trials, they're very different SNPs, but also they're different across populations. We've also observed that in our study and also published studies, generally the SNPs that are identified as small effect SNPs. So we acknowledge that there are genotype by environment interactions that the 1 to 5 scoring scale is not picking up. So moving forward, now that we know that CBSD is a complex trait, the next thing that we're trying to work on is to do a meta-analysis study for all the GWAS studies that have been published. That is data from Nzuki, Kayondo, and my study, to make that one study and see which genomic regions would show up. And then in that, we hope to identify, candidate gene, move forward to develop CASP markers for CBSD resistance breeding. Then another thing we're trying to do is we're trying to expand the phenotyping methods, because we understand that we're not capturing all the interaction between all the components that happen for CBSD to be in place. So we are expanding to include virus titer. That's, to go specific to how these virus species behave with the plant, the virus-plant interaction. We're also trying to include imaging to study root necrosis, to try and expand how much information we can get about the disease symptoms. And then we hope that that will be helpful to identify genomic regions that are associated with cassava brown streak disease. So I would like to thank my committee that's made up of John Luke, Michelle Heck, and Kate Perry, the John Luke and Soros Lab, and the Root Crops Department at the National Crops Resources Research Institute in Uganda. I'll take any questions. Thank you.
We have a question in the chat.
Oh, so Walter is asking if there are any non-dominant resistance genes against cassava brown streak disease. The answer is no, we do not know any.
I have a question. did you notice any environmental characteristics that explain some of the variable genetic effects you saw?
So one is the locations of where we test the material, right? So one is a high disease pressure place and another is a low or moderate disease pressure. And what we've noticed is that since we depend on natural infections, so that means the whiteflies have to transmit the viruses for us, we tend to see that in some locations, one virus species is more dominant than the other. So that could explain all these inconsistencies.
Any other questions from the room? All right, well, yeah, let's thank Leah.
Thank you.
So my research is kind of focusing on the G by E problem. And we know that correcting for environment can enable better selections in plant breeding. When we talk about G by E, we can think about multi-location effects that come from just disparate geographies, but we can also think about the local environment, so things within a single field, either from soil heterogeneity or soil elevation, that sort of thing. And so in the past, we know that design randomizations can help correct for these kinds of local errors. And more recently, there's been like new statistical approaches that capture the interdependence between neighboring plots, so that plots that are closer to each other have correlated or interdependent. So in this research, we're using the genomes to field hybrid maize experiments that are planted at Musgrave Research Station here. And we're using this to look at those local environment effects that I was talking about. Now, we want to see if we can improve genomic prediction. So we have to establish some baseline genomic prediction models. As I'm sure you all are aware, here I'm just defining a model G, where we have a random effect U, which follows, its variance is defined by this genomic relationships. So certain hybrids are more related than others. And we compute G from genome-wide markers. We can improve on that by taking into account spatial corrections. So here I'm defining two models, G plus 2D spline and G plus AR1. And what these models do is they add an additional random effect, and that random effect is structured to account for plots that are closer to each other will have that kind of interdependent effect. Now we can apply that spatially corrected model. Here I'm showing the two-dimensional spline effects and applying it to grain yield, ear height, and grain moisture in 2017. These heat maps show a field level view, and we can see similar effects, spatial effects between grain yield and ear height, with kind of an inversion for grain moisture. Looking at 2019, again, grain yield and ear height show kind of similar spatial patterns. And we saw that again in 2020. And these spatial effects, they can account for up to like plus or minus 25 bushels per acre as illustrated. But the question is, can we do better using UAVs? And so we flew a UAV over these field experiments in 2015, 2017, 2019 and 2020. That's the data I'm using. And we used this image breed program to define the plot, kind of the plot level polygons onto the aerial images. And image breed lets us then like extract phenotypes for each plot. And there's a lot of phenotypes you can look at. Kind of neural networks are like a really interesting method for getting features out of images. And also from the drone, you can get these, it's called a digital surface map. So it's like estimated height. And those are all very cool things maybe for future research. But what I looked at was just NDVI. And NDVI's this ratio of near infrared and red. And it's a useful phenotype because productive chlorophyll reflects in the near infrared and absorbs in the red. And also the camera we're using is well suited for collecting this. And so if I show here in 2020, on the x-axis is days after planting, and on the y is just the mean, the average NDVI for these 15 hybrids. We could see consistent like NDVI trajectories for each hybrid, starting at the vegetative or the growth, early growth phase, kind of plateauing during the grain filling, and then senescent, like dry down and senescence at the end. And so this was like promising, but and potentially useful in itself. But we wanted to try to improve genomic prediction, and so we thought a two-stage approach could work. So in the first stage, we just look at NDVI, and from NDVI, we look at those local environment effects. So again, the two-dimensional spline or the AR1 spatial effect. And a separate alternative model is to use the random regression, which gives us a permanent environment effect. And the random regression is good because it can fit many time points in a single model. And it leverages the fact these are repeated measurements on the same plots. And so from the first stage, we end up getting these low NDVI local environment effects across time. And if we look at what those NDVI spatial effects, how they compare to grain yield, here I'm showing a heat map of grain yield itself, of the spatial effect of grain yield, and of the spatial effect of NDVI at 38 days after planting. So you see a similar kind of spatial pattern between those. And if you look at kind of the whole field experiment, I mean, the whole growing season, from 38 days after planting up to 133 days after planting, you see that the grain yield, the spatial effects for grain yield correlate pretty highly with the spatial effects of NDVI, even early on. So at 38 days after planting, we saw 0.5 and then up to 0.7. So that summarizes the first stage. But in the second stage, we wanted to include those NDVI local environmental effects, and we looked at two different approaches for doing this. So again, here we're adding a random effect, UP, so that random effects variance follows this L matrix, which is like for all the plots in the field, like how related are those plots to each other, and L is like it's a correlation of those NDVI spatial effects. But the second way, which is more simple, is just to put a fixed effect. So H average here, which is just an average of all those NDVI spatial effects. But you can think to go further and separate H into different ranges. So like you have H1 going from kind of the early part of the growing season, the middle, and the late. So we looked at those models, and we looked at heritability and genotypic effect estimation. So heritability, you know, is the additive genetic variance over the with the residual variance. But genotypic effect estimation is where we separated the field into its replicates. So we defined 2 replicates here. Then you run one of these 2-stage models on each block, and then you see how correlated the genetic effects are to each other. And we think this is a really useful metric for determining if the spatial correction is actually working. And so in conclusion, we saw that the 2-stage models tend to improve these metrics, and but we need more years to really validate this. And I'd like to thank the Robbins and the Mueller labs, and if you have any questions, be happy to answer them. So how early in the season can you start scoring from the drone? So really, I think about 30 days after planting, you can start to see the plants well in the images. Since we fly like at 20 meters, so you know the resolution's not super great.
Can you fly lower or not?
You can, but the battery life isn't like you have like 30 or 40 minutes of battery, and if you fly lower, then it'll take longer. So
Right. Okay. Cool. Thank you.
Sure, thanks.
You had every other year data. Do you have more data, or was it not done those intervening years?
Yeah, so Jim asked if I had like 2018, 2016, and so on. Yeah, we have that data, but it wasn't working too well. And I've focused on these 4 years.
Looks good. Yeah, thank you for the introduction. Today I'll be talking about my progress in gene expression profiling and correlation with structural variants to understand the link between genotypic and phenotypic diversity, specifically in tomato. So each organism differs in their genetic sequence, and this variation, or also variance, can range in length from just like 1 base pair to several hundreds of kilobase pairs. And thereby, anything that is larger than 50 base pairs is called a structural variant, or abbreviated SV. Examples for structural variants are something like inversions, translocations, or fusions, but also copy number variants, CNVs, such as insertions or deletions, duplications of parts of chromosomes, or even duplications of full chromosomes. It has been shown that structural variants make up the majority of genomic differences between species and between different cultivars. And they often also are the causal allele with very large effect size because they're so long. However, structural variants are underrepresented in the predominantly used short-read sequencing, so that even though structural variants might often be the causative effect for specific QTLs, they are not detected in previous GWAS, which identify QTL via SNPs. It was also shown, with the use of, or more increased use of long-read sequencing, more studies focus on structural variants now in plants as well. And those studies have identified that structural variants oftentimes overlap with genes or regulatory sequences, and thus they affect the expression of those genes. And they can influence the expression, for example, by altering the gene sequence itself, by altering copy number of that gene, or by changing the position or the sequence of cis-regulatory elements. So I'm interested in understanding the genetic regulation of fruit ripening and the model organism that is used in looking at fruit ripening, specifically fleshy fruits that show ripening phenotypes or that they continue ripening after you harvest the fruit, are tomato. Tomato are also one of the most important vegetable crops. And we see a quite wide variation in phenotypes in tomato, although the genetic basis is quite narrow. So for all of these reasons, I'm using tomato in my research. Two recent papers, both published in 2020, study structural variants in tomato. The first one on the left by Alongi et al. And they identified structural variants in a diversity population of 100 tomato species. And so structural variants are always called or defined in reference to the Heinz reference genome, which is a modern cultivar. So you can see on the left here, wild tomato species have many more structural variants, while commercial tomatoes or modern varieties have fewer structural variants. And most structural variants are either insertion or deletion. And then the second paper, they looked at, or the researchers looked at structural variants specifically between one wild species and the Heinz tomato reference genome, and then they identified a total of 92,000 structural variants. So our recent goal in genetics is to understand how structural variants influence gene expression. And while those two studies have looked into this a little bit, they mostly focused on the identification of structural variants. So this is where my research plays a role. We want to use the available structural variants from those two studies, and then basically build a resource for gene expression profiles that can be correlated with the available structural variants, as well as with agriculturally important phenotypes, such as phenotypes that are related to fruit ripening and nutritional quality of fruit, specifically tomato again. To this end, I have my major project look at a tomato diversity panel of 177 accessions. So I have all of these growing in greenhouses over the last year. And I'm harvesting fruit pericarp, flower, and leaf material for gene expression analyses. And I'm also looking at different phenotypes such as leaf size, fruit morphology, and nutritional compositions of the fruit. For a second project, which is a smaller side project, I'm looking at high resolution gene expression profiles for one of the wild species that is part of my diversity panel. And for this plant, I'm specifically looking at 4 distinct cell types of the fruit pericarp. So this map shows where tomatoes are grown worldwide and mostly identifies or indicates where the 5 tomato species are from that are part of my diversity panel. Those include Solanum tasmaniae and Galapagense, which are from the Galapagos Islands, wild species. And then the third wild species, Solanum pimpinellifolium, which is the only red-colored wild species and is the progenitor of our commercial tomato lines. Then as per commercial lines, I included Lycopersicum variety cerasiforme, which is an early domesticated tomato line, and then Lycopersicum variety Lycopersicum, which is our big fruited modern tomato. As I mentioned on the slide before, I harvested leaf material, flower material, and then fruit material at 3 different developmental stages, which are mature green, which is when the seeds are mature, but the fruit is still green. Breaker stage, which is when the fruit start accumulating carotenoids, and ripe stage, which is defined as breaker plus 7 days in my study. And I harvested the pericarp, which in this diagram here you can see is the green colored tissue. Okay, so all the samples are frozen in the freezer and step by step are being processed for RNA and different metabolites. This image here shows a kind of a collection of some of the tomato fruit, and it's specifically interesting because it shows the different phenotypes, including color, size, and form. And although we don't have gene expression data for fruit yet, over the last summer, I looked at carotenoid profiles with with a high school intern over the summer. And I'm not going to go into the details of the carotenoid profiles here, but we did see variation between the different accessions, which is really interesting. And once we get the gene expression data, we can correlate structural variants not only with the gene expression, but also with like nutrient compositions. Similar to the fruit, we can see a lot of morphological variation in leaf shapes, specifically between, but also within species. And to quantify this variation within leaves, we sent leaf material to the Nelima Sinha lab at UC Davis, where her graduate student, Caro, did like mathematical measures on these samples to get like a quantitative analysis of leaf shape. Then also over the last summer with an RAU intern, we looked at gene expression data from leaves that we had available for 36 tomato accessions. And we also had the structural variant data from the genomes from the 2 studies that I mentioned at the beginning. So the first thing we did was to map our RNA expression data to the reference genome to get gene expression data for the 35,000 genes in tomato. And then we looked at structural variant presence or absence in the 36 tomato accessions. And we started by looking at structural variants that overlap with the genes. And within these 36 tomato accession genomes, we got 5,500 structural variant gene pairs. So where the structural variant is within a gene. To narrow down this number, or to specifically look for SV gene pairs that are present in at least 5 accessions and absent in at least 5 accessions, we filtered these 5,500 and got to 3,500 structural variant gene pairs. Then when you do like a differential gene expression analysis, you usually have 2 groups. You have like your wild type and you have a treatment, like a normal and a treatment. And in our case, we split the accessions, which is why we needed at least 5 with and at least 5 without the presence or absence of the structural variant. So we split those accessions into those with the structural variant and those without the structural variant. And then did differential expression or like significance test for all of the structural variant gene pairs that we have. So from this list, we found 271 statistically significant structural variant gene pairs. So all these 271 showed differential or significant differential expression for whether they had or did not have that structural variant. And from this 271, 103 genes overlapped with a set of literature-curated genes that play a role in leaf development. And the most significant of those structural variant gene pairs actually showed a significant impact on the leaflet phenotypes in the leaf quantitative measurement study. So it showed significant impact over all 5 of the principal components that were used for this mathematical study. And we could also see in photos that we took of our plants that those leaves without the structural variant had a more uniform aspect ratio of the leaf, and those without with the structural variant had more like serrating and lobing density. The structural variant itself as a 143 base pair insertion into a tomato gene that encodes a 2-oxoglutarate and iron-dependent oxygenase superfamily protein. And I don't have more information, unfortunately, about the structural variant or the regulation, but this is something I'll be looking into over the next few months. So just quickly talking about my second Smalley project, which is where I said I'm looking at high-resolution gene expression data, specific for this one wild type, which is part of my diversity panel as well. The leaf or the cell types I'm looking at are within the fruit pericarp, and they are the outer epidermis, collenchyma, parenchyma, and vascular tissue. And I'm also looking at 3 different developmental stages, similar to the diversity panel. I'm looking at mature green, breaker, and breaker +7 stage. And so how do you get to those different cell types? I'm basically, I have to prepare my samples. I cut off the pericarp. The fruit are quite small, so they fit into a mold. They are directly embedded in a medium, frozen, and cryosection onto specific microscope slides. And then with those microscope slides, I go to a laser capture microdissection microscope, and then cut out the specific cells that I want. I have to do RNA extraction on those cells, amplification, and then I can sequence their RNAs. So here you can see a picture from my sample under the LCM, under the microscope. On the left, you can see a photo of the autofluorescence. You can see the different cell types. And on the right, you can see my sample under bright field. You can also see the different cell types. But specifically here, you can see how I cut out an area of the leaf. And this is like on a PET membrane. So it's like a specific slide that I can cut my cells out. And I have not, or at this point, I've only done a few trial slides to see how the laser works and how well I can cut my samples. But I will hopefully soon get to the point where I can cut out my specific cells and then do the RNA sequencing on it. Okay, so altogether, I could show that with my project, I can correlate structural variants with gene expression and phenotype data. And that those structural variant studies can provide insights into the genotype to phenotype relationship. Ultimately, I would like to look into more structural variants that specifically in like the UTRs and in intergenic regions, because so far I only looked at those within gene sequences. I'll also call more structural variants on genotypes that have not been called for structural variants in the 2 studies that I mentioned. And most interestingly, I'm excited to look at gene expression data from fruit, and then specifically look at ripening phenotypes and the effect of structural variants on those phenotypes, both in the pericarp as well as like the single cell types. Okay, and with that, I would like to thank everyone in my lab, my committee members, collaborators, greenhouse staff, and my interns, and you for listening. And I'm happy to take questions. Yeah, Travis.
Great talk. So for the SV gene pairs, are you looking at only exonic sequence or just coding sequence, or do you look at intron SVs as well?
So the question was for the SV gene pairs, if I'm looking at like exons versus introns or the promoter regions. So far, we've only looked at structural variants that are within, I think, exons and introns. We have not looked at promoter regions yet, but that's something that we're specifically interested in to see if there's like structural variant effect on gene expression through promoter regions or other cis-regulatory regions.
This has been a production of Cornell University, on the web at cornell.edu.